My name is Dr. Cassandra Juran, and I am a research scientist with the Bone and Cell Signaling Lab at NASA Ames Research Center. And today I will be presenting my Plus10 talk titled Engineering the OME, in which I will discuss my vision for the potential for omics, investigation, and genetic engineering in space research over the next 10 years. I'll also show data from a series of studies our laboratory has conducted focusing on the implications of engineering a single molecule and how it changes cell, tissue, organism, and community responses from control conditions. Organisms are basically biological machines built by executing programs written in biologic code. It's an overly simplified analogy, but appropriate for the purposes of this talk to compare our genetic makeup to computer code. And again, for the purposes of discussion, let's use the binary computer language consisting of two variables, a 1 and a 0, strung together into meaningful signatures as an analogy for nucleotides on DNA. From these two integers, all computers, smartphones, data repositories, and recording systems are derived. Additionally, the function of hardware is managed by instructions written in this simple code. Biology works in a very similar manner, with a series of 4 variables rather than 2, accumulated in engineered patterns on DNA. Start and stop code initiate and terminate translation of RNA and subsequent transition into protein. The proteins then organize into more complex structures and alter the function in the body. Genetic engineering is not a new field, but the technology to implement a software engineering methodology into the complex genetic computer system is an advent of the last few decades. Some events significant to the development of genetic coding are, first in 2010, Craig Venter's team wrote the genome of a bacterium from scratch and incorporated it into a cell to make what they called the world's first synthetic life form. Later in 2012, the CRISPR-Cas9 system was published as a tool to enable cut-paste omics editing. Later, the commercialization of the CRISPR-Cas9 complex opened the capabilities of genetic engineering to the non-molecular biologist. And then most recently, and most controversially, in 2018, twin girls whom had the CCR5 gene edited before the in vitro fertilization were born in China. These and other studies have sensitized scientists, politicians, and the general public for the implications of genetic tampering. This is where my supposition for development in space research for the next 10 years lies. Spaceflight offers a unique environment for the application of genetic manipulation, either permanent or transient. We can utilize omics technologies for the monitoring, maintaining, and improvement of astronaut health. To support long-duration missions beyond the protection of LEO, we need a system that can counter unforeseen genetic mutations due to stresses accumulated in response to radiation, unloading, isolation, limited nutritional variety, etc. It's my suggestion that in the next 10 years, the importance of researching the implications at a cellular, tissue, organism, community, and even an ecosystem level will need to be considered and leveraged for humanity's outreach beyond LEO. But we also need to recognize how small variabilities can cause awesome changes, and no example is as powerful as that of ocean temperature changes associated with climate shifts. The advent of the industrial age could hardly have guessed the impact of energy consumption's increase of carbon dioxide concentrated in the air and oceans. The accumulation of CO2 leading to increases in the world's ocean temperatures of an average of a single degree Celsius over 150 years. But that 1 degree has resulted in bleaching and death of greater than 60% of the world's shallow water coral reef communities. So before we open the prodigal Pandora's box that is genetic manipulation, we need as thorough an understanding as possible of the implications at all levels of biologic organization. And that's hard to do in a global environment because, like the current coronavirus, change spreads quickly and beyond the boundaries of our control. So, spaceflight, as I said earlier, is a uniquely isolated environment where genetic changes due to stresses are likely, if not unavoidable. And as such, research into implementing omics monitoring and maintenance may prove a powerful technology for maintaining Earth norm health in astronauts on journeys to Mars, or dare I say, even beyond. An example of leveraging omics observation and genetic manipulation that our lab has been involved in is the study of the cell cycle regulator molecule CDKN1A, P21. An mRNA survey of STS-131 mouse bones identified CDKN1A as a highly upregulated gene of interest. P21 is a cell cycle regulator with function in cellular arrest, stem cell differentiation, and DNA damage response. The figure to the right shows that ground control samples have minimal protein presence in the periosteal surface, whereas with spaceflight, the protein's presence is significantly upregulated. Interestingly, when the CDKN1A gene is knocked out of a mouse model, the mouse exhibits heightened regenerative abilities. Shown in the figure to the left, we see ear punch wound healing on the bottom panel, that over the course of 35 days, the ear punch has closed and you have a scarless healing, which is similar to amphibians with their regenerative capabilities. If you take bone marrow stem cells from these knockout mice, you find that the cells are hyper-responsive to mechanical stimulation, resulting in elevated proliferation and differentiation during an osteogenic protocol. Uh, this provides evidence that we might be able to tune regenerative responses of certain cell types by manipulating the expression of P21. Increasing our experimental resolution on the bone marrow stem cell population to single cell level, we're able to observe that CDKN1A expression is not universally mechanoregulated, but instead only committed osteoblast cells, which have progressed beyond the progenitor state, respond to mechanical stimulation with CDKN1A suppression. This is highlighted by the pink arrows. This data demonstrates that cell type is critical when considering genetic regulation of regenerative mechanisms. When considering pharmacological regulation of gene presence, a recent publication assessed synthetically knocking down CDKN1A P21 during wound healing and was able to show that the utilization of silencer RNA suppression of P21 or P21 deletion was able to return a healing ability to aging mice that matched younger controls. This proved the efficacy of targeted single gene of interest regulation in regenerative response. Before concluding, I would also like to discuss how the utilization of omics tools can broaden research platforms by inclusion of atypical animal models. The animal kingdom has many unique creatures with unique capabilities. The jellyfish, for example, has been called immortal due to unexhausted stem cell-like turnover. The microbiome of the Hawaiian bobtail squid can be leveraged to understand symbiosis and organ development, and coral reefs can be observed to understand community dynamics. Spaceflight investigations have probed regeneration using the Iberian ribbed newt on at least 17 missions. However, the genome for this species was only sequenced in 2019. Our lab is in the process of analyzing sequenced historic regenerating tail samples from a Photon M2 and M3 missions to better elucidate the molecular mechanisms of regeneration in space. And interestingly, we've identified a potential P21-like molecule which is expressed strongly in the dedifferentiating stump of a regenerating tail. Thus, using this unique animal model, we can learn more about P21, its role in regeneration, its role in adaptation, and its role in evolution. My final thoughts on where genome and transcriptome engineering will be over the next 10 years, or where it's going, is that first, omics investigations are going to continue to become more commonplace, even in labs as remote as the International Space Station. NASA will likely continue supporting health monitoring and fundamental science using omics data analysis. Um, Genetic regulation of mRNA expression will likely be considered to counteract significant changes in astronaut health due to spaceflight. And genetic engineering will likely be considered to improve astronaut performance throughout long-duration spaceflight missions, incorporating personalized medicine platforms. Genetic manipulation is an awesome ability that needs implementation slowly and with constant consideration. Long-duration space exploration is a likely source of genetic mutation, Thus, implementation of genetic monitoring, pharmaceutical silencing of genes associated with dysfunction, CRISPR or Cas9 genetic editing techniques to inhibit or delay onset of degenerative conditions during space missions may be appropriate countermeasures. We have the tools, but we need to understand the impacts. And investigations to understand these impacts is where I think research will grow over the next 10 years. Thank you for your attention.